Hello, our project is on gene instability. Gene instability is the increased tendency for genomic errors or mutations that affect cellular division and tumor suppression that is known to occur in cancer cells. This basically talks about how cancer cells are almost always associated with an unstable genome. An unstable genome can lead to a cascade of events leading to increased amounts of mutations occurring in the genome. This leads to the increased likelihood of cancer cells arising and developing traits that favor their survival and growth into a tumor. As you can see from this graph, there are many DNA repair mechanisms that have different functions to ensure that the genome is stable and appropriately structured. The large amounts of DNA repair mechanisms show how crucial it is to maintain the integrity of the genome. Damage or malfunctions to any of these DNA repair mechanisms can be deleterious to the stability of the genome.
MGMT is a DNA repair enzyme that stands for O6-methylguanine DNA transferase. This repair mechanism functions to protect DNA from alkylating agents. Alkylating agents damage the DNA by adding a methyl group to a nucleotide in the DNA. MGMT specifically responds to a guanine base that has been incorrectly methylated. A methylated guanine base can be mutagenic and ultimately lead to mutation. In the first diagram, we can see that a methyl group has been added to the O6 site of the guanine nucleotide. An active MGMT enzyme will take the methyl group and restore the guanine to its normal unmethylated form. A guanine base typically pairs with cytosine. However, if the oxygen is methylated, that hydrogen bonding site will be inhibited and the guanine will bind with thymine. Therefore, MGMT is required to promote gene stability. According to studies surrounding MGMT, both hyperexpression and silencing of the gene coding for MGMT has been linked to oncogenic properties. Studies are still being done on the significance of this pathway, therefore most of the information on MGMT is speculative and requires more research.
Epigenetics is an inherited modification to the genome. One such mechanism for modification is methylation, which is generally associated with silencing a gene. This is done by inhibiting certain transcription factors and polymerase from binding to the DNA sequence. As shown in the graph, methylation at the promoter sequence of MGMT inhibits the expression of MGMT from being expressed, while the absence of methylation allows the DNA to produce MGMT proteins.
Another plausible method on how MGMT methylation leads to cancer is that guanine and thymine are paired, mentioned on slide 6, which shows a similar pattern of KRAS mutation. RAS mutation is responsible for 25% of all the tumors, and among these RAS mutations, 90% is caused by KRAS mutation. In short, 22.5% of the entire tumor is caused by KRAS mutation. According to Carpino's research, in the case of pancreatic ductal adenocarcinoma, KRAS mutation not only leads to cell proliferation, but also affects the tumor microenvironment and regulates metabolism, which benefits the tumor cells, which also requires further research for details. Another study by Nakagawachi demonstrates the correlation between KRAS mutation and the hypermethylation. Among 244 patients, 42% demonstrated hypermethylation, while 36% among the total showed KRAS mutation. We cannot generalize it yet, since it requires more research studies, but throughout the articles we've gone through, on average, more than 80% of the patients had a relationship between hypermethylation and KRAS mutation.
According to this graph pulled from a study showing the expression of MGMT, the increased expression of MGMT has shown to be correlated to GBM or glioblastoma. The mechanism for how is still being researched. We can postulate that this is due to poor methylation of the promoter, and so there's a higher amount of MGMT being expressed. Once again, the importance of the stability of the genome is shown to be critical and why genome instability is a hallmark of cancer.
One of the methods of treating cancer is chemotherapy. It uses cytotoxic drugs, and TMZ is the most standard treatment for brain tumors like glioblastomas or Hodgkin's lymphomas. A brief explanation of the mechanism of TMZ is that the alkylating agents deliver methyl groups targeting DNA bases. After this process, if cytotoxic lesion O6-methylguanine is removed by MGMT or tolerated by the mismatch repair deficiency, the patient resists TMZ and other alkylating agents. The effects of these drugs differ based on individuals. According to Dr. Hartley, more than 60% of glioblastoma patients are MGMT positive, and for them, the survival rate is only 15% after 2 years with treatment, while the MGMT negative patients' survival rate is 50% after 2 years of treatment. Thus, it is crucial to find an alternative treatment that is more efficient for the patients. We found Dr. Harley while we were doing research for TMZ. He helped us understand the principle how MGMT positive has more resistance towards TMZ. He is a professor of chemical and biomolecular engineering and a research team leader at the University of Illinois. He develops complicated biomaterials that replicate human tissues and organs. Currently, he is developing a mutated version of TMZ that cannot be removed by MGMT, leading to cancer cell death.
In addition to gene instability, MGMT has been speculated to influence different hallmarks of cancer. If MGMT is silenced in relation to the KRAS pathway, the KRAS gene will be more prone to alkylating agents and therefore mutation. Since KRAS is a significant oncogene, its mutation may lead to cell proliferation. Some studies have found that hypermethylated MGMT may also contribute to the mutation of p53. Similar to that of the KRAS pathway, MGMT silencing may contribute to p53 mutation, which will directly allow the cell to evade apoptosis. However, the information is mainly speculative. The oncogenic potential MGMT holds makes this gene of particular interest to researchers. Although research is still being conducted on this particular gene and its pathways, there appears to be a clear relationship between either the overexpression or the lack of MGMT in cancer cells. With more information to be found, new treatment or chemotherapeutic methods may be discovered. MGMT is established to be a potential chemotherapy agent, and learning more about the relationship between the state of MGMT methylation and chemotherapy agents such as TMZ may ultimately prove to be useful. And here are our sources.